Hello, everyone, and welcome to today's webinar titled From 50 to 500 Genes, How to Scale to Comprehensive Genomic Profiling While Reducing Costs, Time, and Labor. This webinar is part of the 13th Annual Clinical Diagnostic and Research Virtual Event. I am Megan Pascal of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by QIAGEN. For more information about QIAGEN, go to www.qiagen.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located on the left of your screen. You can also participate by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar from our lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speaker, Dr. Beate Litzenburger, Global Product Director of Oncology and QIAGEN Digital Insight. For a complete biography on our speaker, please visit the Presenter tab from the menu at the left of your screen. Dr. Litzenburger, you may now begin your presentation.
Thank you so much for the nice introduction, and thank you all for joining us today. QIAGEN is the leading provider of genomic and clinical knowledge for scientists and clinicians using augmented molecular intelligence. What that means is that Ph.D. scientists, engineers, and ontologists blend artificial intelligence methods and human intelligence to curate the world's largest and trusted source of molecular knowledge, the QIAGEN knowledge base. While machine learning techniques help to rapidly prioritize and extract, index, and seed evidence that inform our subject matter experts, every piece of information is manually reviewed and further manually processed to curate complex biological processes and clinical evidence to achieve deep insights with precision. The QIAGEN knowledge base drives powerful analytics, visualization, and reporting tools that enable the researchers, the clinical scientists, and the data scientists to excel their cutting-edge research and clinical decisions. So why is content key to adopt comprehensive genomic profiling and even whole exome or whole genome sequencing? The burden to interpret novel variants in larger panels, such as the comprehensive genomic profiling, whole exome or whole genome sequencing, does not increase linearly with the number of additional genes. Instead, it increases exponentially because not only one must contend with more variants, but these variants are also less well characterized, and there may be incidental findings, and they may interact with each other, impacting the prognosis, diagnosis, and treatment response. Thus, pathologists and clinical labs are challenged to maintain turnaround time without jeopardizing quality for patient management. So according to an independently conducted survey, the most significant bottleneck in the NGS workflow is the length of time spent analyzing data, with over a quarter of researchers surveyed report that performing NGS data analysis requires more than a week. And then QIAGEN Clinical Insights platform eliminates this bottleneck and allows your lab to adopt new panels, scaling volume, and deliver with confidence accurate patient reports to your clinicians. And we will talk in the next 30 or so minutes on how QCI can successfully help your lab to scale or launch your genomic profiling service. So this is to orient ourselves here a little bit. The QIAGEN Clinical Insights platform consists of flexible software tools enabling a seamless, fast Q2-report solution for any panel and any sequencer. There are various different secondary analysis solutions that seamlessly connect to QCI's tertiary oncology analysis software, QCI Interpret One. And today, what we're going to do is we're going to dive deeper into QCI Interpret One and specifically how you can identify incidental findings and interpret co-occurring variants. But before we do that, I will provide a brief overview of QCI Interpret One. So how does QCI Interpret One work? QCI is a cloud platform that offers flexible analysis of single or multi-samples from any NGS tests, ranging from comprehensive cancer panels, whole exome sequencing, to RNA-seq to deliver interpretation for any cancer type. At a high level, how do you generate reports using QCI Interpret One? There are different data file imports. The most commonly data file import that we prefer is the VCF file that can minimally be contextualized with the patient's disease type, and that allows for accurate display of disease-specific content. So in the first step, the variants are prioritized based on the most clinically relevant variants. Each variant will be automatically classified according to the AMP and the ACMG guidelines and associated with appropriate treatments and trials. In the second step, you will retrieve ready-to-use variant and disease-specific summaries without the time-consuming step of writing them by yourself. In addition, our experts provide you with a second opinion on the variant classification to increase the confidence in the accuracy of your classification. The QCI platform provides the flexibility for customization of the workflow, as well as a flexible customization of your final report. So the QCI offers the flexibility to utilize the user interface and API endpoints to interact with the platform. The APIs allow connections to your LIMS system from which you can upload new tests or samples. You can search for tests and their status and receive various exports, such as the PDF report or XML files with structured variant and disease-specific content. While the user interface allows variant scientists to manually review the content and make edits, users can substantially increase turnaround time and reducing any hands-on time by automating various steps in the interpretation process, ranging from just the upload to the user interface or the upload all the way to fully automating the variant assessment, the drug and trial selection, or all the way up to retrieving interpretations and a final draft report, all the way up to downloading the information back into your LIMS system without even utilizing the user interface at all. So how does QCI display a patient's molecular test? So this is a variant list of an NGS test of an AML sample. Each row represents an alteration detected in a tumor tissue. The different color codes, such as red and pink, display the QCI computed pathogenicity of an alteration. And the bottom center indicate the QCI computed actionability classification. The smaller badges on the right top corner are informational badges and provide an initial clue of what information is available for this variant, such as the variant is sensitive to therapies or has prognostic implications. This view basically provides a broad overview of the patient's molecular profile. You can also transparently review the prioritization cascade and review the filtering parameters that your lab has set. You can walk up and down the filtering cascade to review variants that are filtered out at each step if needed. The transparency is useful to also identify low allelic fraction acquired resistance mutations or diagnostic mutations that may not necessarily pass to the bottom of the cascade. However, this transparency allows you to still report those low allelic fractions even if you jump to a different level in your filtering cascade. For each alteration that is detected in a patient's profile, QCI provides a one-stop shop organized in several expandable sections for you to review the detailed information about the alteration in context of the user-submitted phenotype. In the interest of time, I'm not diving deeper into each of these sections, but I want to point out specifically two major sections. The first section is the functional impact section that is here open in the bottom of the screen. It provides you with a written summary of the molecular function. You can also access the curated references via hyperlinks. And on top of that, you'll be able to review the predicted biochemical impact from various prediction tools. And the combined knowledge allows you to confidently understand the functional impact and relevance of this mutation. The second section that is front and center in the variant details page of QCI is the assessment section, which displays the computed actionability and the pathogenicity criteria. So here I want to focus on the actionability for this variant. All tiers that are applicable will be shown to transparently indicate the actionability options for this alteration. Some variants have therapeutic actionability as well as a prognostic actionability. And then for each tier, the underlying clinical evidence can be viewed, which provides transparency in the sources that support each tier. And if you're thinking about therapeutic actionability, you can then dive deeper into the treatment section where all applicable treatments and clinical trials are pre-selected for the draft report. Again, the references can be opened so that you can review the underlying evidence and applicability to your patient or your region. One thing to note is that the user still has the full control over adjusting the actionability criteria by removing or adding a criterion, which you can see here at the bottom of the screen, and making adjustments to the final assessment of the alteration. In this section, basically, you report the pathogenicity and actionability for each alteration. The last step is the report building step and allows you to generate comprehensive reports in minutes without the time-consuming step to write interpretive comments by yourself. So in this review and report page, QCI Interpret 1 displays all variants that should have been reported, reportable variants that you can see here at the bottom of the screen. And then for variants that are clinically and biologically relevant, you will see a highly structured interpretive comment with every sentence backed up by references. The user has editorial control to refine the content for the report, add or remove a piece of information, change wording, or otherwise revise the content. And these changes can be saved and will be easily accessible in the future. And within minutes, you can generate an oncologist-ready report, which provides a comprehensive overview of the patient's molecular profile. The expert, variant, and disease-specific summaries are provided for biologically and clinically relevant variants. And the first page of the report not only provides an overview of potentially relevant approved treatments, potential resistance, but also provides a note to the oncologists of any interactions or any potentially relevant guidelines that could be applicable here for this patient in context of that disease. So let's go and discuss more details of the content that you can access through QCI. So users of QCI Interpret 1 take advantage of over 20 years of expert curation of genomic and cancer-related literature aggregated in the QIAGEN knowledge base. And that is combined with over 10 years in performing somatic variant interpretations. And thus QCI provides your lab with access to the world's largest curated knowledge of biological and clinical variant and disease-specific information. QCI's platform has processed just for oncology alone over 700,000 patient cases and over 3 million cases in total, counting in hereditary cancers and carrier screening. With a growth rate of over 100,000 new characterized variants per year, it's not just the largest knowledge base, but also the fastest growing knowledge base in our space. Why should you trust QIAGEN's genomic knowledge? Behind the scenes sit an excellent team of experts who are oncologists and Ph.D. scientists. QIAGEN variant scientists work very closely together with practicing oncologists from a range of subspecialties, such as the solid tumors, the heme space, neuro-oncology, who apply direct clinical experience to the interpretation and ensure clinical relevance and accuracy. As of today, QIAGEN's knowledge base contains over 21 million curated alteration findings, and that's derived from manually curated literature, drug labels, guidelines, and clinical trial databases combined with publicly available databases. All curation and transformation of unstructured data from the literature is performed according to the same ontology and data models that ensures basically semantic consistency among all these resources that are combined. So the resources combined bring a wealth of usable, trustworthy information regarding the biological, clinical, and disease relevance of an alteration to view. And for transparency, this information comes with appropriate citations within the application, as I showed to you previously. Users of QCI can also access report-ready variant disease-specific interpretation summaries with detailed interpretations of gray areas in complex scientific situations, and thus assisting the user in report writing and helping to further shorten the turnaround times. Those summaries are available for all kinds of biomarkers, including SNVs, MDLs, fusions, the copy numbers, MSI, TMB, as well as LOH, cytogenetics, and protein expression. The variants are described specifically in terms of their molecular impact and are classified according to the AMP guidelines published in 2017. The variant and disease-specific clinical evidence covering potential therapeutic options as well as prognostic and diagnostic significance is reviewed and approved by a team of practicing medical oncologists. QCI Interpret 1, as I mentioned before, provides also disease and gene-specific relevant treatment guideline summary, as well as an analysis of multivariant interactions between variants in the same gene or between variants in different genes. And these interactions are basically selected based on the level of evidence and pre-curated based on the literature. Now, what differentiates the QIAGEN knowledge base from other databases? Because the content from the literature and databases is transformed into machine-readable units, QCI can perform dynamic computation and enables automatic updates of variant actionability, pathogenicity, functional consequences at scale for every variant as new evidence is added daily. With QIAGEN updating significant volume of data weekly, you can see we have about 45,000 different changes in our content findings every week. You're able to stay up to date and confident in your decisions, in your variant interpretation decisions. Now we will dive deeper into specific topics that I've prepared for this webinar today. Since a substantial proportion of genes included in large tumor panels confer a heritable predisposition to cancer, tumor genomic profiling is associated with the possibility of identifying incidental germline findings. And the American College of Medical Genetics and Genomics recommends the return of secondary results for a subset of 73 genes, but other genes with evidence of clinical utility are currently emerging. For pathogenic sequence variants in cancer susceptibility genes identified on tumor-only sequencing, it is often unclear whether they are of somatic or constitutional origin. And such variants are sometimes referred to as tumor-detected pathogenic variants of unknown origin or variants of unknown origin. So tumor genomic profiling are estimated to detect a germline pathogenic variant in 4 to 12% of patients. This means that every fifth oncology patient has a germline pathogenic mutation. And this graphic here shows the percentage of TCGA cases carrying a pathogenic or likely pathogenic variant in each of these 33 cancer types. On the right is the number of pathogenic or likely pathogenic variants in tumor suppressors, oncogenes, or other genes in each of these cancer types. Recognition of these germline findings or germline variants is essential not only for optimal care for a patient with cancer, but also it initiates a cascade of genetic testing in at-risk family members who may also carry the familial mutation. So many cancer panels include cancer susceptibility genes among the list of clinical actionable genes, including high penetrance genes with published medical management recommendations. The list that you see here is not complete, but should illustrate that the actionability needs to be related to the current cancer of a pathogenic germline mutation. So, for example, if you're taking a BRCA1 germline pathogenic variant in an on-tumor setting, such as breast or ovarian cancer, may influence the choice of chemotherapy, eligibility for targeted agents, such as PARP inhibitors, and or primary surgical management. On the other hand, if a BRCA1 is found in a bladder cancer, it is considered to be off tumor, since germline BRCA1 mutations do not confer elevated risk to bladder cancer. In cases of on-tumor detected pathogenic variants, there is a higher probability that the mutation comes from a germline rather than it's somatically acquired. The identification of incidental findings during tumor profiling is quite complex, making it even more challenging to meet your turnaround times. By Bioinformatic tools can computationally predict whether a certain variant is germline in origin. The prediction is based on several factors, such as allele frequency, population frequency, so nucleotide composition, and potentially damaging effects, and the presentation of a variant in a database. Now, bioinformatics is just one piece of the puzzle. It is quite common to detect, for example, TP53 mutations in different human carcinomas while the somatic tumor profiling is performed. However, the general germline conversion rate is only 1%, while this percentage is a bit higher in the on-tumor setting, which is about 2%. That means that the detected TP53 mutation is usually somatic in origin unless it's detected in a patient with a personal or family history of Li-Fraumeni syndrome. So in such cases, critical appraisal of the variants of the patient's clinical presentation and family history needs to be used to adjudicate whether this may be a patient with a diagnosis of a hereditary disorder such as Li-Fraumeni syndrome. So due to the complexity of identifying the origin of a mutation, bioinformatics analysis should be combined with the manual evaluation of personal and or family history of a cancer, the on-off tumor association, evaluation of existing founder mutations, available literature to assess the affected individual's inheritance patterns and confirm the causative association with the phenotype using the ACMG classification criteria. So why is this important for a diagnostic lab? Every fifth patient will be diagnosed with a potential germline pathogenic variant. The potential hereditary germline variant may predict future cancer risk, and it may be predicting response to cancer-directed therapies. Also, the detection of pathogenic germline variants is often a critical step in the initiation of a cascade of other genetic testing, potentially also of relatives, and evaluate the risk of other affected family members. So how can QCI help identify potential germline mutations while analyzing a cancer case? QCI contains content for over 4.9 million characterized germline variants. There are PubMed references available for every published germline variant in over 300 cancer risk-associated genes, including variant and disease-specific associations. In addition, HGMD and ClinVar are included as additional evidence to determine disease-causing variants. And to determine if a variant is indeed pathogenic and associated with a hereditary phenotype, QCI supports the computation of all 28 ACMG criteria. So let's look at an example in QCI. This is an example of a frameshift variant in a high penetrance cancer susceptibility gene BRCA2, and with a allele fraction of 45% and not present in the somatic database like COSMIC, it is suspect of being a pathogenic germline variant in an on-tumor context, in this case, ovarian cancer. One can further manually inspect the literature and ACMG criteria. So QCI provides in the reported clinical cases for ovarian cancer a visual clue if the variant has been detected in other affected unrelated individuals. One can open the bibliography where the NLG statement of an article states if the variant is of germline or somatic origin, and if it's primarily observed with the tumor type of interest. Now, you may have noticed in the relatively high nomad frequency in the header, and it's particularly high in the Ashkenazi Jewish population. Reviewing the ACMG criteria shows that it's indeed an established common pathogenic founder mutation. The transparency in the ACMG criteria allows the user then to quickly review all the evidence to determine a potential causative relationship to ovarian cancer. You may also notice that in this blue box here is that the pathogenicity has been automatically calculated for hereditary breast and ovarian cancer. QCI utilizes the ACMG 73 gene list to alert users of pathogenic variants causative of associated phenotypes and provides the pathogenicity classification for the associated phenotype if the allele fraction of the mutation is above 40%. So as this could be indeed a BRCA2 germline variant, which can confer to elevated risk of ovarian cancers, user can then set the assessment to secondary findings to be called out on the report. And when you generate a report within QCI, QCI flags those pathogenic variants suspected of germline origin and gives a recommendation for confirmatory germline testing. So as key takeaway, a sizable portion of pathogenic variants in cancer susceptibility genes that are observed on tumor-only analysis are of germline origin. QCI provides in-depth information of detected variants and suggests potential germline origin where appropriate, prompting further manual inspection of criteria for potential additional testing and or genetic counseling. Clinical decision support tools should aim to distinguish somatic versus germline origin by assessing the variant allele frequency, the presence of a variant in databases, literature references describing the origin of a variant, relevance to the presenting tumor, and utilizing all 28 ACMG criteria to point to a causative relation to the with respect to the on-tumor setting. So let's dive deeper into the second topic, which is how QCI delivers evidence for the effect of co-occurring variants on prognosis and diagnosis. The reason I picked this topic is that the rapid increase in cancer gene mutations by comprehensive genomic profiling reveals a high level of complexity in which mutations co-occur. Clinically, it is important to include co-mutation genes in cancer panels and to adequately annotate the co-occurrence of mutations that may affect further clinical management. So the example that I picked here to highlight the complexity is the interaction between FLT3-ITD and NPM1. The prognostic effects of FLT3-ITD and NPM1 mutations in AML are well established. It is shown that FLT3-ITDs with wild type NPM1 confers a poor prognosis, whereas NPM1 without FLT3-ITD confers a favorable factor. These findings likely have clinical impact and making it of utmost importance to detect and take into consideration the existing of these co-mutations. Now, the prognostic evaluation of AML is not only dependent on the combination of FLT3-ITD and NPM1, but also on the FLT3-ITD allele ratio. So a FLT3-ITD to wild type ratio of above 0.5 confers an independent adverse impact on event-free survival as well as event-free survival, as well as overall survival. And when you take the NPM1 mutation status into consideration, NPM1 mutation positive AML with FLT3-ITD allele ratio below 0.5 has a favorable prognosis. And in those scenarios, the allogenic hematopoietic stem cell transplant in the first complete remission period is not always actively recommended. So how does QCI handle the scenario of FLT3-ITD and NPM1 co-occurrence? So here you can see an AML case with FLT3-ITD and NPM1 mutation in the variant list. You can verify the FLT3-ITD status by quickly confirming the function if it's indeed a mutation that has the insertion in the right mucosal membrane domain. And because while QCI's bibliography provides references to the new guidelines and articles describing the effect of co-occurrent mutations, these complex scientific situations are very well handled by our QIAGEN's expert interpretation summaries, which includes such details when building your report so you can confidently assess the prognostic outcome and risk classification of patients with AML. So what you see here in the summary statement is that the interactions take into considerations the high allele ratio or the low allele ratio, and pointing then to the user in this case of a favorable prognosis, in the case of a low allelic ratio of FLT3-ITD and AML. So in summary, the co-occurrence of mutation may be associated with clinical outcomes or may support the accurate disease diagnosis. The interpretation of co-occurrence is challenging, especially when thousands of mutations appear in one clinical sample. And while I've shown you today only one example, QCI's bibliography provides references to the newest guidelines and articles describing the effect of coexisting mutations, and the expert summaries take into consideration not just the two mutations and the effect of these two mutations on the prognosis, diagnosis, or treatment, but it also considers all the details around it, such as the allele ratio. It could be also FISH or IHC results that are taken into considerations. The last topic I want to touch upon is rather emerging, as most labs perform tumor-only analysis. However, several academic institutions have implemented a tumor normal analysis workflow, and liquid biopsies are gaining broader traction, having the advantage of lower procedural risk for patients and the possibility for longitudinal tracking. A paired approach to analyzing tumor and germline sequencing data would be expected to improve the efficiency and accuracy of distinguishing somatic mutations and germline variants, and thereby facilitating the process of variant curation and therapeutic interpretation of somatic reports, as well as the identification of variants associated with germline cancer predisposition. So this approach is being adapted at various academic institutions and provide advantages to tumor-only sequencing. First is the detection of unforeseen hereditary variants, including the cancer risk mutations. Second is the detection of somatic mutations to guide targeted therapies. And these can be subtracted from the germline variants that occur in every cell, leaving only basically quiet somatic mutations for interpretations. And the third is that it's possible to identify potential clonal hematopoiesis-related variants. Of course, several challenges exist regarding the implementation of paired tumor normal analysis in patient care, including technical, practical, and most of all, cost-related considerations. The second use case of analyzing multiple samples is longitudinal tracking, which can be possible with the broad adoption of liquid biopsies. Liquid biopsies can be used to detect initially the targetable mutations, but then subsequently can be used for response monitoring and identification of actionable resistance mutations in patients. So how does QCI handle multi-sample upload for simultaneous analysis? QCI can upload up to 200 samples, where you basically drag and drop your samples to the uploader, and then subsequently these samples get validated and uploaded to QCI's sample management. Now, if you aim to initiate a tumor normal test, that's what you see here on the left, you basically drag your cancer samples in one box and the control samples you're in the other box, and then continue with your analysis. If you aim to initiate a longitudinal analysis, what you can see here on the right is you would basically drag all your cases to the case upload, but leave the control sample upload empty, and then continue with your contextualization of the test with the disease. Within the application of a tumor normal analysis that you can see here, you can see that every case, every variant is represented with its allele frequency. You can see that you have two columns, a case and a control column. And in the first highlighted box, you can see that this variant has been clearly identified as a somatic mutation only. But at the bottom, in the case of the CHEK2 and the MSH2 mutation, these are both present in the tumor as well as in the normal tissue, and therefore are germline variants. If you analyze NGS tests of cancer samples from different time points, as this is the case here, the variant list displays within the case column the allele fraction that were detected at the different time points. For example, the ATM mutation was present only once in the initial sample, but not in the other 3 samples that are shown here. Whereas if you go to the FANCD2 mutation, this mutation was only present in the 3rd sample, but not in the other samples. So this view allows the tracking of allele fractions of mutations over time to track disease progression, to monitor therapy response, and to investigate potential acquired therapy resistance mechanisms. In summary, simultaneous analysis of multi-samples can be done in QCI to help discriminate between the germline and somatic context of a variant, track disease progression by analyzing molecular profiles at different time points, and perform therapy response monitoring and investigate therapy resistance mechanisms. And with that, I would like to close with an overall summary. QCI enables you to confidently interpret NGS variants from any NGS panel. QCI can help you to accelerate turnaround times by providing dynamically computed classifications, access to written interpretive comments, and the automation of the workflow to scale to higher volumes. Lastly, the report layout is fully customizable to comply with your brand recognition. You have easy editorial control, and API capabilities allow you to integrate the workflow within your lab system. And with that, I thank you for your attention, and I would be happy to take any questions. If you would like to request a demo, please Google QCI for Oncology or QCI Interpret 1 to access the website and for requesting a demo. Thank you very much.
Okay, thank you, Beate, for that interesting presentation. We will now take the rest of the time to answer any questions that we have. Again, please use that Q&A panel on the left side of your screen to submit questions. It looks like we've gotten several already, so we'll go ahead and get started. The first question that we have is, do you retain a history of the variant classification, and are you able to search for variants that we have reported on?
Yeah, that's a good question. We do retain the history of the variant classification in two different sections. One is when you're interpreting the variant itself. In the variant detailed section, there is a history of previously clinically observed variants from your lab that you can review. You can review who annotated that from your lab, what was the classification, when it was done, what was the cancer context, and you can review any additional notes that the previous variant scientist has left. The other place where you can review the variant history is in our variant directory. You are able to search there by the gene, and you can find then all the variants by gene, and you can see across the whole gene how often you have interpreted and you have seen that specific variant and access it through that interface as well.
Okay, thank you. The next question that we have is, what kind of variants can QCI Interpret 1 process and interpret, and what data files can be uploaded?
So the first, let me answer the first question first. What kind of variants can a QCI Interpret 1 process? So of course, the small variants, SNVs, indels, small duplications, but QCI is also able to process the structural variants that you can encode in form of a VCF format, for example. Those are fusion, rearrangements, small copy numbers, focal copy numbers, as well as large copy numbers, large copy number deletions. For the fusions, we do display, for example, the breakpoints where the breakpoints fall in, if it's in an intronic region, an exonic region, which exon it's affecting. And for the copy number, we key off the genomic coordinates, and that means that depending on if there's one or multiple genes falling into that region, it can be displayed as a cytogenetic change to indicate that multiple genes are affected by your copy number gain or loss. And then we interpret other TMB, MSI, and in the upcoming release, we'll be also able to handle homologous repair deficiency status as well. The second question was, what data files can we upload? We do typically like to take in the VCF file, but do support for the TSO 500, for example, the TSV, CSV files that are part of the standard second analysis output from an Illumina pipeline. So those are the three most common data files that we can intake, but happy to work with any of you to intake other forms, as long as we can parse out the genomic coordinates and is part of like a proper 37 or 38 genome build.
Okay, great. The next question that we have is it possible to configure or customize the final report?
Yeah, the report can be customized at two different levels. First of all, it's the content that goes onto the report. I think I showed you that you can edit the report content. That's up to you how you want to edit the content. And then the second point is the layout configuration. So we do have some basic layouts configuration changes that you can do by yourself. But if you want to comply, for example, with your own brands, your own coloring, your own font, we have a report service that works with you to configure and customize the report the way you like it.
Okay, thanks for the information. Next up is, how are the oncologists involved in the process validating the clinical evidence in the interpretation summaries?
Yeah, that's a good question. So when we are curating the literature for its clinical relevance, meaning summarizing the clinical studies or prognostic studies, any clinical studies that have been published, as well as the guidelines and curating the guidelines, we will not publish this data within our database and route this through our internal tools to the oncologists, and they have access to the tool so that they can basically review and make changes. They are asked to make, of course, changes in a timely manner so that we can publish the content as likewise in a timely manner. And that ensures basically its clinical applicability, especially when the evidence is maybe not as well defined as in some cases of, let's say, BRAF V600E in melanoma, but of some of the emerging biomarkers and maybe some of the less evidence that's available even in the guidelines, then we really heavily rely on the oncologists to help us make sure that it's still clinically applicable.
Okay, thank you. We have time for a few more questions. So the next one is, can you select clinical trials in QCI based on the patient's location?
Yes, you can. So there are two ways to select clinical trials. The first selection is based on when you upload your data. You can specify a zip code, a city, a country, and that will be used to display the appropriate clinical trials based on your region. And the second option is within the user interface itself. If you would like to go to the clinical trial section, and then you can select your country and or your city, as well as a specific radius around that to narrow down the clinical trials that are applicable to you.
Okay, thank you. Again, we have time for just a couple more questions. If you do have questions, please keep submitting them. If we don't get to your questions today, we'll follow up with you by email. The next question that we've received is, can you integrate the results seen in QCI-I1 into a local LIMS system?
Yes, we talked about this briefly at the introduction of QCI Interpret 1. We do have APIs that allow us to connect to your local LIMS, and from which you can then access different API endpoints to QCI.
Okay, great. Next is, can I issue amended or corrected reports after a report is finalized or signed out?
Yes, there are ways to do that. So if a report is signed out in QCI, it has a status of final. So you would need to basically open a final report and and then once you open that up, basically you have full access to what was reported, and you have also access to what may be new. So, for example, if you open up the review and report page of a final report that was maybe final half a year ago, and you reopen it, you could see that QCI would potentially gray out specific clinical trials that are not applicable anymore. And you can see what you have previously classified and how you have previously classified the variant. And then, since QCI always computes the latest evidence, you can always see how QCI computes the variant today. So if you do that and want to then issue an amended report, basically you have a case report field that allows you to make changes there, and also add comments that this is an amended report, and then reissue that report and finalize it again.
Okay, great. I think we have time for one more question. So the last question that we have time for is, can we search and pull all patient reports with certain genetic variants?
Yeah, that's a good question. So we do have a variant directory which allows you to basically search all reports for a specific variant. We currently don't have an export functionality. However, we do offer this as part of a service where we can customize the fields for you of how you would want to export the data and in which detail. So there are ways to basically pull all patient reports. We do save all of them as part of clinical standards, so that we can enable this type of export for you.
Thank you, Dr. Litzenberg, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, QIAGEN, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand for 2 years until November 9th, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.